you so much. Sorry. This is so good to have everyone over here. I would like to thank the AATM chair, Dr. Anand, and vice chair, Dr. Sangeeta, to have this excellent presentation on the plasma protein therapy. And we will be speaking today on plasma collection, quality control, and challenges. And we have a few panelists who are going to present today, and we are going to have a good discussion on that. So this is a new topic for all of us, or some of us. And even if we've had some sort of training in our plasma processing, we are going to delve deeper into how we are fractionating the plasma and how we are using it, and how it's an essential therapeutic today. So I would like to request Dr. Sangeeta if she can, speak for or provide her comments on this session. And I would like to invite her. Dr. Sangeeta, is it okay for you to? Yeah,
thank you so much, Dr. Asa, that you have taken so much pains for this panel discussion. And I'm actually very grateful to the entire panel. And just a few words about the plasma protein therapy, plasma collection and all. The plasma fractionation, it might be a very small part of this great, huge biotechnology industry. But still, for us, it is a very important part because we are providing a lot of therapeutic benefits to the patients through these components that are being prepared here. And the discussion that is going to take place today is along with a very elite panel, and the lead is also a very elite lead, Dr. Asa, and she would be leading this panel discussion. And I'm really grateful to all of you. Thank you so much, and most welcome to this panel. And over to you, Dr. Asavri, to take this forward. Thank you so much.
Thank you so much, Dr. Sangeeta. It's wonderful to have met you through this forum and to learn so much from each other. And I think this is a very good platform that AATM has created to learn from each other what everybody's countries are doing, what we are doing, where we have been trained. So I would also suggest, I do have a slide for each panelist, but I would also like them to, speak a little bit more about what their facilities are doing, because ours is a very small group and we have time so we can talk about, each other's expertise, what we need, how we can, move forward in this group. At the beginning, I would like to just introduce the topic. So if I can begin, I have a little bit of a presentation to introduce today's topic. So I can introduce that and then we can move forward. Thank you so much. As a housekeeping, I would like to ask everybody to please mute their mics, and we can, once we have the panel discussion, then we'll have our mics open. So thank you so much. If anybody has any questions, we will ask the questions at the end of the everybody's presentations or the panel discussion. Thank you so much. So today's topic as a group from Plasma Protein Therapy is plasma collection, quality control, and the challenges that we face. Sorry. Can you see it now? Yes. So I have 20 plus years of global leadership experience. I'm a globally validated subject matter expert for clinical trials, quality management systems, regulatory compliance, gap audits. I'm now a faculty for designing and conducting trainings and medical educations in different countries such as UAE, US, and many of the other countries. I work now as a consultant for cell and gene therapies through many of the companies that I represent. I'm the elected member of CTSCC, that is the Cell Therapy Subsection Coordinating Committee at AABB. AABB has recently been branded, so Association for Advancement of Blood and Biotherapies, that's its new name, and we have been having many projects through AABB. I'm the co-chair at Asia Pacific Group, and I would invite everybody of us to join that group to get to know more about what we are doing. If you're an AABB member, it's free of cost to you, so it's at no cost. I'm also the co-chair at the Cord Blood Banking Company. If you've had inspection, I get your international variances because we sit through that committee. The current CT standards are being reviewed and written, so I'm a part of that committee, and I represent the global experts for cell and gene therapy in the AABB. I'm the chairperson of AATM since the last 3 years now, 2.5 years almost, and I'm the chair, speaker, moderator, and panelist at various conferences, and I've published publications and written books. Sorry, I've got to do this at two ends. So these are my disclosures. These are some of the companies that I work with. And now that we have, so these are some of the things that we will be speaking about. So what is plasma fractionation? What is the process of plasma fractionation? What are the risks associated with it? And the regulation and guidances if we decide to move forward with plasma fractionation, and the areas of specific relevance. Why is it important? What do we really need to do? Every one of us is already aware that we have to deal with the donor screening and how we can take care of that. There are new updates on that as well, especially after COVID, after Zika. There are many other things that we need to be careful about. We also today talk about the epidemiologic surveillance and why it is required for the donor screening, eligibility of donor screening, then the various methods that we would be talking about, then the processing of the plasma and the appropriate collection method. I mean, why is it important for us to choose the appropriate collection method? And then we'll go on to our panel discussion today. So when we look at plasma fractionation, what is plasma fractionation? Plasma is harvested from donors for the downwards processing to plasma fractions. And currently it is an industrial process where it is used to isolate therapeutic plasma proteins, which are also known as the fractions, or the process is known as the fractionation. And the process of fractionation actually breaks plasma into individual proteins or plasma fractions, which can be used to treating many conditions. It is different from the whole blood fractionation, where, taking the whole blood and then we break it into 4 parts, and how we are going to discuss it in the later slides. Currently, there are about 20 different plasma protein therapeutics, which are used as an essential class as the only available option, especially in the life-threatening diseases or injuries, trauma, infections, congenital deficiencies, and immunologic disorders. So if we look at the process of plasma fractionation, the starting material is all about pooling the donor plasma. And there are around 10,000 to 50,000 donations which are required for the industrial processing of plasma fractions. And there are different volumes. So there might be a smaller facility and a bigger facility. So some of the bigger facilities where the industrial processing is happening, they pool a lot of plasma. And there's around 23 to 28 million liters of human plasma are fractionated each year. And currently there are around 70 factories of processing or industrial processing of plasma fractions. Some of us are actually at the downstream because we are the people who are collecting it. We collect the we collect the plasma, and then we send it to these factories where it is further processed. And these are very sophisticated industrial processes, which are performed under highly aseptic conditions in licensed facilities. They also require good manufacturing practices and the regulatory environment, because when we are pooling 10,000 to 50,000 donations or even lower volume, we need to have a good perspective on the whole chain of production. So there needs to be a good governance for the whole chain of productions. They are operated in compliance with CGMP or good manufacturing practices, and they need good quality assurance principles, such as the quality management systems, what are required to run these kind of sophisticated plants or factories. So what is a process of plasma fractionation? Most of us currently use the modern plasma production technology, and it is largely based on the ethanol fractionation process. There are being novel methods which are being looked into in the last few years, which are being used to improve the product purity to enhance the recovery of immunoglobulin G or isolate new plasma proteins. So even if we know how to process the plasma, but slowly we are learning more and we are designing new methods to look into how we can improvise on the product, how we can enhance the recovery of certain fractions which are more important, and getting into more basics of these fractions on how they can be helped, such as alpha-1 protease inhibitor or Wald-Brand factor or protein C. Obviously, when we are pooling so much of plasma together, especially it could be from different centers, different donors, different countries, we do have many risks which are associated with the process of plasma fractionation. And these major risks are mostly related to the transmission of the blood-borne infectious agents. And this is also the reason why we need a good regulated professional environment where fractionated plasma products can be manufactured with the lowest risk therapeutic biological products. So screening is very important. The eligibility of the donor is very important. And we are going to talk more about this. And we use dedicated viral inactivation. There are some new modern technologies which have now come out. And the removal of these, blood-borne infectious agents. So there are some treatments which are available with it. It has been implemented throughout the production chain for the last 20 years, but currently we are looking more towards how we can improvise it. This process is actually done to ensure optimal safety in particular and not exclusively against, HIV, Hep B, Hep C, and other viruses that can affect this product formation. So the important point for us, especially when we are having this panel discussion and today's webinar is all about regulations and guidances for plasma fractionation. So what do we actually need? From each of us, we need a plasma master file where it should contain all the information about the process, including the collection. So we need a good donor history. We need how much of volume was collected, who collected it, and is it traceable? What screening was done? What were the different tests that were performed? What was the different quality control that was performed on it during the processing of the fractionated plasma product? And today our panelists are going to discuss some of these details with you. It is also the part of the marketing license of plasma products. So we are liable or we are responsible for this end in the chain of custody or chain of identity. And now there are various requirements for the collection of plasma for fractionation, Pharmaceutical Inspection Convention and Pharmaceutical Inspection Cooperation Scheme, which is also called as referred to as PICS. US FDA has a very clear guidance on this, and WHO has provided some good recommendations and guidelines for plasma fractionation. Now, when we look at it, we have to look at certain areas of specific relevance, especially again, when we are doing it for our downstream plasma fractionation. The collection requirements of plasma for defractionation is actually different for the whole blood as well as the fresh frozen plasma. And we need to understand that, especially the temperature where it has to be stored, the temperature at which it has to be logistically sent to these fracties. These are different. And in a regulated environment, especially countries like us, plasma for fractionation is collected by licensed and registered blood establishment. So we have to be registered with the government, registered with the proper regulatory authority, and these are inspected by the relevant national regulatory authorities or NRAs as we call it. Due to the safety concerns, the plasma fractionators conduct audits. So if we have a tie-up with a company in US or in Europe, we do have audits from them, and we verify that the contractual plasma collection and quality and the safety measures are met according to their requirements. So Europe has a different requirement of regulations, and are we complying with that? Plus, every plasma fractionator or every person who is doing this fractionation has a requirement, because they're looking at a particular fraction and not all the 20 fractions, which I just mentioned previously. So the areas of specific relevance, they include procedures for donor screening and donation testing, labeling, documentation, traceability requirements, and especially the handling of blood and plasma. The quality requirements of the plasma fractionation most of the time are related to two important aspects. One is the inactivation and removal of blood-borne infectious agents, and then adapting the best practices or methods for the purification of various fractions. The steps involved in donor screening are candidate donors are provided with educational materials, and most of the time it has to be in their own language. So each region, each country has to develop these materials. They have to undergo a medical interview to establish the absence of risk or signs of infection, and to prove compliance for a donation of plasma for fractionation. And if there are potential donors who are presenting a health hazard, then they have or they should be able to avail to exclude themselves. And medical information is acquired and archived, especially as a part of the plasma master file. This has also become a very good tool to provide the epidemiologic surveillance, especially hemovigilance. For, it helps us to establish the background level, prevalence, incidence, different infectious markers. We can also go to the genomic sequencing of these, viruses, and we can provide the early detection. We can look at stringent donor screening processes and the requirements for additional testing procedures. Eligibility of donor screening is very important, and we'll be touching more when we have the panel discussion. The donors eligible to donate plasma for fractionation are individuals who meet the donation criteria. And this is based on age, donation frequency, what your NRA has prescribed, what your fractionator wants, as well as the WHO recommendation and WHO guidelines. Do not present the risk factors of blood-borne infectious agents, and they comply with requirements defined by your plasma fractionator and NRAs. In most situations, the eligibility of whole blood donors and apheresis donors may overlap, but then we have a different protocol might be adapted, which can be used, which is higher for plasmapheresis donors, which we need to differentiate between these two. And the eligibility criteria takes into account scientific information about the risk of transmission of infectious agents. As we know, most of the kits that we use are already approved by each regulator or US FDA or many other regulating agencies in your country. However, there's a small error or small minor chances that we can infect, or the infectivity of or transmission of these infectious agents is high. So we need to be careful on that end. And a special criteria may exist for the collection of, for an example, hyperimmune plasma, which is used to make hyperimmune IgG. And for this, we may need to have the donor's immunization and minimal antibody titer to be represented there. Now, there are different methods that I spoke about, and the collection of plasma is most of the time done by plasmapheresis. At minimum, the red blood cells are returned to the donor while plasma is retained and collected in a container. It could be a bag or a plastic bottle. The typical plasmapheresis duration depends on the number of cycles and generally lasts from 35 to 70 minutes. We also decide how much of the volume we are going to, go through these or, protocol to be followed. The apheresis plasma is also called the source plasma. It is collected from donors through a process where blood is removed from the donor. An anticoagulant, generally with 4% sodium citrate solution, and is immediately separated by physical means. It could be centrifugation or filtration, or a combination of both. And again, the volume here is 450 to 880 ml, depending on the country's regulations. And we also look at the byproduct, which is the platelet pheresis, also called as concurrent plasma, a procedure which is used primarily for the collection of platelets. Now, both recovered and apheresis plasmas are suitable for the manufacture of the whole range of fractionated plasma products. This is the plasma collected from the whole blood. Again, we've already discussed this, but whole blood donation is about 220 ml And this depends on the donor's hematocrit. It also depends on the fraction that you're actually trying to obtain. So for processing, currently about 35% of the plasma fractionated in the world is obtained by centrifugation of whole blood, or which is recovered plasma, and 65% is obtained by apheresis. And again, the collection, processing, storage may affect the plasma quality, such as impact on recovery of the most labile proteins, such as F8. In particular, risk of activation, which may lead to generation of plasma proteases, should be avoided. So how can we minimize coagulation, reduce the activation of complement or fibrinolytic systems? And to better preserve the integrity of recovered plasma and limit risk of activation, we should remember to look at good mixing of the blood with the anticoagulant solution. And the duration of the collection ideally should not exceed 15 minutes unless and until the cycles are required. The temperature variations of the blood should be avoided. Now, again, these are some of the examples. Like for apheresis plasma, it contains less IgG. So if we need more IgG, we should look at either concurrent plasma or plasmapheresis. Protein content and quality of fractionated proteins is usually not affected by the apheresis system. So residual cell content differ upon the type and configuration of the cell separation devices. So we have Optia, we have different companies. I should not be biased to one company, and we should talk more in general about these different companies where we are using it for apheresis or plasmapheresis. Plasma from membrane apheresis procedures, as does recovered plasma prepared from whole blood, is leukoreduced on positively charged filters. So we have these different filtration methods. Ethanol filtration is one, positively charged filtration is different. And these are the different methods which can impact the quality or the yield of fractionated products. Again, longer processing time before freezing. So if we talk about the F8, whole blood must be further processed to separate cellular components and plasma. So we need to choose the proper or appropriate method, and high ratio of anticoagulant, possible high level of cellular contamination that may release proteolytic enzymes affecting the stability of the coagulation factors. And that is why the mean content in coagulation factors, we need to consider that for the specific fraction that we need to look into. So today we have a panel discussion. Sorry, I think this is where I can stop and we can go to the next presentation. Can I stop sharing? Or can I change to the next one?
That will be great, ma'am.
Okay, thank you.
Thank you.
So I'm just going to put the panelist. Sorry. Are you able to see my screen share?
Not yet, ma'am.
Okay, just a second. Yes, is it possible? Can we see it?
Yes, ma'am. Yes.
So today we have a discussion on plasma collection, quality control, and challenges. And today we have, I'm moderating the session. Our panelists today are Dr. Chandani Damika, Dr. Hina Qureshi, Dr. Nova Hippi, Dr. Mustapha Allavathi, and Dr. Kalyan Roy. And I beg your pardon at the initial listing if I did not pronounce your name correctly, but this is a great opportunity to get to know each other. I have already introduced myself, so I'll go to Dr. Chandani Damika Sediva Ratne. She is from Sri Lanka. She has received her MBBS from Sri Lanka. She's holding a diploma in transfusion medicine from the Postgraduate Institute of Medicine, University of Colombo. She's an MD in transfusion medicine. She's a clinical fellow in transfusion medicine from Singapore Blood Service, Health Science Authority, Singapore. Currently, she's holding a consultant transfusion physician NBTS position with the Ministry of Health. She's also the head of Anuradhapura Cluster Regional Blood Center, the teaching hospital at Anuradhapura. She's the secretary of Sri Lanka College of Transfusion Physician, a co-chair of AATM, and visiting lecture faculty of medicine, University of Rajaratha, Sri Lanka, and a committee member, Executive Council of 2022 in Anuradhapura Clinical Society. Thank you so much, Dr. Tamika. It's wonderful to have you here. Would you like to open your mic? I would request all the panelists to open their mic. And then if they want to speak something more about themselves, Dr. Tamika, before I go to the questions, it's wonderful to have you here. And how is Sri Lanka currently? How's the weather and your country?
Yeah, we are so far okay. Yeah, thank you, Dr. Asha, and the organizing committee giving this opportunity to being a panelist in this important panel discussion of the plasma fractionation and the quality control. Sure. This is my questions regarding. Shall I continue, Dr. Asha?
Yes, please, please, please. I just wanted to know, what are the programs which are being implemented in Sri Lanka for collecting plasma? Do you have any plans to, start this fractionation process? And any idea you can talk more about, these questions that we have posed to you?
Yeah. In the first questions regarding the programs implemented in Sri Lanka for collecting plasma. First of all, that I will tell about our service. It's a National Blood Transfusion Service. Is a centrally coordinated blood donations via the cluster management system. Each cluster center is headed by the consultant transfusion physician. They supervise the donor selection, recruitment, and post-donor care. Manpower, equipment, consumables, SOPs, and protocols are provided from the National Blood Center, as well as the performance monitoring at each centers by monthly statistics form. 100% voluntary donations established since in the Sri Lankan Blood Service since 2004. About 90% of whole blood donations comes from the mobile blood donation camps, and remaining 10% comes from in-house donations. Cluster centers and other large-scale blood banks provided with a PHI, Public Health Inspector, to organize in the mobile blood donation camps and recruitment of the donors. And PHI and blood bank staff work with the community organizations to organize in the mobile blood donations to collect in the blood. Especially in the religious organizations and other community organizations also incorporated regular blood donation programs into their annual activity plan as a regular organizers. Regular awareness programs are conducted in each centers for mobile organizers, and then we will take the feedback from the organizers at the end of their each mobile sessions. That is helpful to improving our donor base, as well as to improve the that system. So in addition, for promoting the donations every year that will celebrate in the World Blood Donor Day in all blood banks to encourage the volunteer donors to promote their blood donation. For the collection in the National Blood Transfusion Service concerned, annually our collection around 400,000 units whole blood, all from 100% voluntary blood donations. That 100% component productions go from the whole blood donations. Usage concerned about 50% out of the annual collections that we use, and other remaining 50% we export for the fractionation. This is about my first questions. That what do you need more about this? Any questions regarding these programs in our National Blood Transfusion Service?
Thank you so much, Dr. Damika. I also wanted to know when you said that it is exported, do you have a distributor or do you have a supplier that collects from you and then supplies it back to you?
Yeah, we supply as a supplier.
Very nice, very nice, very commendable, Dr. Damika. And uh, they supply back to you or at a cost, or how is this
No,
done in
no,
Lanka?
not we didn't get that back. We only supply send the excess plasma for the fractionation for the foreign company.
Okay, very nice, very nice. Thank you so much, Dr. Damika. If there are any questions for each other, we'll keep it at the last end, if that's okay. Dr. Damika, thank you
Yeah.
so much for participating and giving us such a good overview of Sri Lanka and letting us know how we can do this thing. Okay, I'll go to the next panelist now, if it's
No, that
okay
is the
for
other
you.
one. Sorry, for the
No,
challenges
please,
concerned
yeah, yeah.
in our facility in the country for donor selection and suitability and eligibility. That we have a very big problem with in the collection. The donor population, they are increasing. Their elderly population is increasing in our country. Also that is also one big problem is this increase in trending of the non-communicable diseases like diabetic and hypertensions. That because of this economic crisis, majority of the populations, their lifestyle changes also will affect to our collection. In the upward trends of the sexually transmitted diseases in the population, especially in the MSM activities, majority of the our donors are male, about 74% and the 26% of female donations. Both of them have same hemoglobin cutoff value used for both male and female donors. With the current crisis, economic crisis that increasing in the population with anemia, especially in the females, that will badly affect. It will be a badly affect to our female donations with the same hemoglobin cutoff value. Our all the donors in our service that they are registered in the online database. Lack of computer hardware and the internet coverage is a challenge, especially in the remote areas. And then each donors are individually counseled by our qualified medical officers prior to the donation, providing that is also the manpower is provision of the manpower is becoming a challenge. And then the blood bank information system, this is BBMS, that implemented in all the blood banks, that does not have access, but does not have access to the donor health records. Most of the minor medical conditions are deferred due to lack of proper records. Temporary deferral donors in different reasons are not followed by the available system. This is one of the challenge for us. Confirmation of the TTI test in algorithms are very complicated, and then it takes time. That time consuming. Donors are not allowed by the system until TTI investigations are done. The recently 3-year deferral is enforced after visiting the malaria endemic areas countries, and since 2021 up to now there is no challenge, but I think in future when travel restrictions reduced after COVID, it will badly affect to our blood collection. Yeah, that's all.
You have really highlighted some of the challenges that we are all facing, and I think somewhat similar. There might be a few more or less, but I think overall that is the trend that we are seeing even globally. So this is really of concern. And I think after COVID, it has imposed a lot of, especially resources, connectivity, mobile, how do we provide counseling for them? That is also a important task. Thank you so much, Dr. Chandani. I'll go to the next this thing now. Uh, Thank you so much again. Actually, next on the discussion was Dr. Hina, but she has a presentation. So can I go to Dr. Nova with everybody's this thing, if that is okay?
Yeah, sure.
Yes, sure, ma'am. Yeah.
It's okay.
Hello, Dr. Nova.
Hello.
Dr. She is MBiomed, MPH. She's the medical faculty, University of Indonesia. She holds a master degree in biomedical sciences, medical faculty, University of Indonesia, and a master degree in hospital management, medical faculty, University of Gajah Mada. She is holding the current positions as Senior Blood Project Officer, Australian Red Cross, Tsunami Response Team, Banda Aceh, Indonesia, Head of Blood Transfusion Services in Sipto, Mangunkusumo National Referral Hospital, Jakarta. She's an internal auditor and preparation committee of JCI accreditation, Head of Blood Services Division and Central Blood Establishments of Indonesian Red Cross, Jakarta, Indonesia. She's a lecturer at D3 of Transfusion Technician, Jakarta, Indonesia. And the awards that she has received is the Best Unit in Sipto Mangunkusumo Hospital Accreditation. She's been an ISBT Fellowship Award winner, just like Dr. Aikat here, and NZ SBT Fellowship Award. So very nice to have such an excellent, panel, Dr. Chandani, Dr. Nova, who will be sharing her, vision, her current focus in Indonesia. Thank you so much, Dr. Nova, to have you here. And these are the questions that we have posed to you. If you can let us know your country's position or facility participation in performing the plasma processing and fractionation and some of the challenges faced in Indonesia. Thank you, Dr. Nova. Over to you.
Thank you, Dr. Asa, for the kind introduction, and thank you, AATM, for giving me the opportunity to participate in this event. I have been given two questions. The first one is about whether Indonesia has already participated in the plasma processing or fractionation. Actually, before Indonesian Red Cross has already participated in plasma fractionation by sending plasma to other country with fractionator. The first one is to Australia, and the second one is to South Korea. But it was stopped during the monetary crisis. And so at this moment we doesn't participate in plasma fractionation, but we are hoping in the coming years we will be able to participate again. The second question about what are the challenges faced in successful implementation of plasma processing and fractionation in our facility. In 1980s, Indonesian Red Cross, like I said before, already sent plasma to the fractionator and made albumin and some IVIGs. But at that moment, the market only asked for the albumin, so the IVIG is not real, was not a popular product at the time. Due to the monetary crisis in 1998, the fractionation program was stopped, and also there is another problem at that time because the clinicians preferred the import products than the one made by Indonesian Red Cross. So at that time, Indonesian Red Cross had so many albumins that had to be thrown away because it was overdue. So that time it was decided we won't send plasma again to another country. Many years later, the concern about so much plasma that has to be thrown away and the higher needs of PDMPs made Indonesian Red Cross think about participating in the fractionation program again. But the requirement to send the plasma was different than before. Yeah, and you already know the geography of Indonesia. It has a lot of many islands, and it makes fulfilling the requirements a little bit complicated. For example, the price of the reagent in the west of Indonesia may differ than the one in the east of Indonesia. Uh, because the cost to send the reagent is high from one place to another. So yeah, the price is different for one reagent and in one place and in another. Hence, the quality of plasma may differ from one place to another. In 2013, Indonesia was appointed by WHO as one of the countries that will be assisted in improving the plasma quality. Indonesian Red Cross started the project by implementing the GMP program in some blood establishment. After that, Indonesian FDA started to get involved, so GMP certification was started. At the moment, there are already 19 GMP certified blood establishments. Most of them was the blood establishment with the highest plasma production, so we could supply enough plasma to the fractionator. To make sure that the supply of plasma is enough in the future years, because the volume needed may keep increasing, Indonesian Red Cross started a regional system by dividing Indonesia into 5 regionals, with each regionals consists of some sub-regionals, and each sub-regionals have one blood establishment appointed as the coordinator in centralization of testing and blood processing, blood supply networking, quality control, referral test, training, logistic, and monitoring evaluation. It will become another challenge in the future because, yeah, so many things to do to make sure that the other blood establishment will have a same quality with the first ones. So at this moment, Indonesian Red Cross has made some plans as the plasma suppliers, but we still have to wait for the government. And right now, Indonesian government is reviewing some regulations and make some technical preparations, so Indonesia could participate in the vaccination program, hopefully in the couple of next years. So that's what I want to report. Thank you.
so
Over to you.
much. Yeah, thank you so much, Dr. Nova. This is very interesting that you all were already being a part of that, and obviously the challenges, the resources that we are dealing with now has changed than what we were doing in 20 years back. But this is good to know, and I think because of your closeness to the Australian continent, it becomes much more important for you to also adapt some of the regulations and some of the requirements for that. So I think this is a very interesting discussion, Dr. Nova. Thank you so much. I will
Thank
go
you.
to the next panelist now, that is Dr. Hina. And then Dr. Hina would actually be sharing her screen as well. So, Dr. Hina, welcome to this panel discussion. Her educational Thank you. Her educational qualifications are she's done her MBBS from Dow Medical College, Karachi, Pakistan. She has done her FCPS Hematology and College of Physicians and Surgeons of Pakistan. I would say you're my alumni as well because I did from CPS India. Quality and healthcare, yes, quality in healthcare and clinical governance, Pakistan Institute of Management, Karachi, Pakistan. She has written many research papers, case reports, publications in many national and international journals. She is part of the Pakistan Society of Hematology, life member, coordinator of Karachi chapter since October 2020, and executive council member Sindh region since February 2021. She's part of the Pakistan Biosafety Association, life member since 2012, and AATM Vice Chairman Pakistan since December 2021. She has many accreditation and surveys that she has participated as head sections of hematology and blood bank at Suleiman Al Habib Medical Group, Riyadh, Kingdom of Saudi Arabia. Then she has been part of Planetary College of American Pathologists of CAP, Saudi Central Board for Accreditation of Healthcare Institution, a BPJCI Healthcare Information and Management System, or HIMSS, John Hopkins University and Aramco survey, and currently she's working as the consultant blood bank, Medina General Hospital, King Salman Medical City at Medina Munawwar. Over to you, Dr. Hina, and these are your questions, but please feel free. I will stop sharing the screen now. Sorry, I will stop sharing the screen so we can go to your.
So, hello from Saudi Arabia and Assalamu alaikum to everyone. My part of my question was the quality control in plasma fractionation. And I will start that what exactly our goal should be. Should it be the disease of the patient or the patient himself or self? And how to provide treatment. As doctors, we should be providing treatment which should be safe and following high quality standards so that safeguarding public from unsafe therapies. With that goal in our minds, plasmapheresis or plasma fractionation from either whole blood donation plasma or plasmapheresis, we should have this in our mind that safety is an important aspect. Along with quality and high standard, safety is an important aspect that should be monitored. So plasma obtained by plasmapheresis or separated from a unit of whole blood donation can be fractionated into various components. And the various components could be albumin, immunoglobulins for IM or IV use, coagulation factors, anticoagulant proteins, protease inhibitors, or fibrin glue, and many more other derivatives that are of medical importance. Quality control is a part of the quality assurance program, and overall it starts from where we are recruiting the donor and where we are where the donor is incoming into this process. So if we have an organization, we have a facility, we have manpower there, we have resources there, are they adequately trained? Are they adequately competent to go through the process of plasma fractionation, starting from the donor entry in the system? So quality assurance in process controls will encompass organization, facility management, documentation system, of course. Supplier qualification is very important at this step because when we are having the bags on board, the equipment, the machinery, the instrumentation, how we are going to fractionate plasma, that is in a later step. But starting from the bag, selection of bag, it should have certain qualification criteria so as to have a quality product and a safe product. When we have donor on the board, donor identification is very important. Eligibility, as Dr. Allavathi has already spoken about, and selection of the donor. Then whether the donor is for whole blood donation or whether he has opted for plasmapheresis, then the adequate time period in between the two donations. That is all encountered, that should always be looked into. At the time of collection, reducing the bacterial contamination with a good venipuncture, maintaining the sterility, sterile zones, everything. And then after the blood has been collected, the laboratory testing for safety, viral testing, and other tests by which we will know that this particular donation is eligible for a safe transfusion further. So at the time of processing, production and fractionation of plasma, good manufacturing practices, and there are various components of this good manufacturing practices for the product, that is our end product. For that particular end product, various fractionation procedures are followed so as to reduce the viral load and inactivate the viral load as much as possible. For certain certain viruses can be filtered out. So we will use the filtration processes, while certain other will be ethanol, a certain will be heat inactivated, or and so on. We should have a very strong IT system for this purpose, because after recruitment identification, if the donor is found at some point in time with the testing that he has to be notified about his testings are positive, how to recall, look back, how to notify, all that are a component of a good quality assurance system. And of course, the storage and transportation temperatures and conditions and criteria that we have to meet. So at each and every step to assure the quality and safety, when we have the final product, we also do certain quality control measures to see if that product is adequate for use or not. And then, of course, all these measures should be audited and reviewed by management and regulatory and monitoring authorities. So what will be the steps of quality control of plasma or plasma-derived medicinal products? General, as we have just now discussed, donor eligibility selection. I'm not going to repeat it. You can all see this. And the TTD testing requirements from plasmapheresis screening for syphilis. For the plasmapheresis donor, the screening of syphilis is important on the day of donation, and the protein component testing is important. It should be 6 to 9 gram per deciliter, and certain other tests like serum protein electrophoresis or any other methodology by which we can quantify the composition of the protein, quantify the proteins. The processing should be within 6 hours. The temperature requirements, as we all know, that it should be minus 20 degrees or below, and it should be constantly within 6 hours of when it has been processed. It should be at minus 18 degrees and below. There should be no evidence of thawing while storage, during the storage. Labeling and relabeling should be adequately monitored and should be timely put on the product. The container, quality of the container, it should be leak-free, no color added, hermetic seals should be intact. The final product should not have any indication of physical change or indication of contamination, microbial contamination. Specific quality control for plasma, for FFP at the initial stage, it is the fresh frozen plasma or the liquid plasma. Every 3 months, measure of factor 8 levels, in at least a statistically derived figure for the particular blood bank in which this is being monitored or quality controlled, but not less than 4 bags each month. So it should be factor 8 should be less than, it should be more than 70, more than or equal to 70%, 17 national units. Similarly, cryoprecipitate should be checked for fibrinogen content, 150 milligram or minimum of 18 national units of coagulation factor 8. For albumin, the storage temperature has to be -5 or colder if it has to be stored in a bulk quantity over a week. But under a week, then there is less restricted temperature control. Heat treatment within 24 hours for 10 to 11 hours at 60 plus or plus minus 0.5 degrees centigrades. It should be heat treated. Stability should also be checked. The color should be unchanged if treated at 57 degrees for 50 hours, and the color can be checked by another albumin which has not been heat treated, and by comparing the color. The concentration of sodium potassium should be 130, 160 for sodium milliequivalents and 2 milliequivalents for potassium. The pH, which is a very important part for, again, viral reduction and viral inactivation, it should be 6.85 to 6.95. Plasma protein fraction, again, the similar as we have already seen in the albumin. The total plasma protein content should be more than 5%, 5 gram per deciliter. The pH should be 7 plus minus 0.3. The immunoglobulins should be checked. Approximately 2 ml of completely processed material should not show. For the heat stability testing, we can use 2 ml of completely processed material, which should not show any signs of deterioration of the plasma product after heat treatment of the whole lot at 57 degrees for 4 hours. The pH again 6.8 plus minus 0.4 should be turbidity free. Storage should be at minus 10 degrees or below. After sterilization, temperature exposure above 32, but not within more than 72 hours. Antibody tests as per standards for different antibodies like diphtheria, measles should give adequate international units, adequate presence of these antibodies. Along with the specific component, general and specific requirements of quality, staff competency, proficiency testing, written agreements, especially with the fractionation plant or a fractionator or plasma between the plasma suppliers and the fractionators. This should state all the quality requirements which should be met by the plasma supplier as well as by the fractionator. Documentation, records, donor adverse reactions, referrals, notification, counseling, all constitute a part of quality assurance. For the second part of my question, which is the challenges faced in performing the QCs, as we've already discussed, and as Dr. Asa correctly said, that more or less we are all facing similar things, and especially in the third world countries, there is we need dedication national regulatory authorities. We need good governance and leadership at national and facility level. The allocated health budgets are very small. There are lack of adequate policies and guidelines, and therefore standardization within the blood banks, within the transfusion facilities, within the plasma suppliers, they are lacking. There are inadequate audits and inability of the collection center to meet the requirements of the regulators. Budget constraints again, and lack of expertise, skilled expertise is also lacking. Starting from the donor recruitment, there should be effective communication skills so as to reduce as much input of those donors who are a potential cause of spreading the viruses to the recipient. This should be looked into, and this is a very, what should I say, it's a very challenging job that training, staff competency at this level is very important, and community level awareness. Way forward is that if we have component therapy, we will have surplus plasma left in our blood banks. And maybe this plasma can be used for the medicinal product derivatives. Cost effectiveness will also follow once we are having the fractionation either at our homeland, or we make contracts with other countries and nearby countries and follow all the quality control procedures. Training and awareness, of course, is the main focus, should be the main focus. Thank you so very much.
Thank you, Dr. Hina. Thank you for such an excellent presentation, and good to have you have those details as well. I did not get into too many details based on, what we were going to add, but thank you so much, because I think quality controls are very important. And as you correctly mentioned, knowing the correct cutoff values, knowing the correct assessment method is also important for what you're looking. I just had a quick question for you. Do you use any UV irradiated mechanism for, removal of? Yeah, sorry, go
Uh,
ahead.
UV, actually in Pakistan, we are still exploring this opportunity of fractionation of plasma. So I'm not very much well aware of this.
Okay, no problem. No, thank you so much. And
And if any of the participants know, they can also speak about it.
yeah, this is why we are going to have a little bit of an open discussion at the last end. Dr. Hina, I will go to the next one so that we have two more presentations. So thank you so much, Dr. Hina. This was wonderful. I'm going to. Can you stop screen? Sorry.
Yes, I will do that. But one more thing that I wanted to just share
Sure, sure.
here is this is apart from the topic discussion. This Pakistan flag that you are seeing, you know that in India and in Pakistan, it was our Independence Days in August 14th for Pakistan and 15th for India. So that is why I put this. Happy Independence to both the country members.
Thank you so much. Happy Independence Day to you too, Dr. Hina.
Thank you.
So I would like to go to our next panelist today. He is from Oman, Dr. Mustapha Allavathi. His educational qualifications are he's a PhD from molecular immunohematology, molecular background of serological D negative in Omani population. He has his BSc and MSc by medical science from Liverpool John Moores University in UK. He also has a diploma in medical science from Institute of Health Sciences, Oman, Associate Fellowship of the Higher Education Academy in UK, and Medical Education Certificate, Dundee University, UK. His current position is lecturer in Oman College of Health, former lab in charge of a tertiary hospital in Muscat, Al Nada, worked as lab technologist, mainly hematology, in Al Nada Hospital, Oman. He has won many awards and publications: Best PhD Poster Award at university level, Best Overall Diploma Graduates Award in Oman, and many publications and manuscripts under review. He is also the vice chairman of AATM Oman Chamber, and he is also the resource person for academic activities at AATM. I would like to ask Dr. Roy if he can share Dr. Mujtaba's presentation. Is it okay, Dr. Roy? And we can also go to your presentation next, Dr. Roy.
Yeah, thank you, ma'am.
Yeah.
I'm trying to share Dr. Mustapha's presentation.
Sure. Until then, these are the questions that we have posed for Dr. Mujtaba, where we see a requirement to implement plasmapheresis centers in Oman to achieve a constant supply of raw material and plasma products, and what are their current challenges to recruit donors in Oman, especially in regards to donor suitability. So we'll just wait for Dr. Roy to uh, share his screen. Are you able to share, or should I stop? I can stop sharing. Yeah.
Yeah, ma'am. If you can stop sharing, then I can try to share.
Yeah.
So
I
shall I
stopped
start?
sharing. Yeah. Yes, please.
Can you see the screen? So, ma'am, can you share? Can you see the screen right now? I have shared already.
Yes.
It is visible.
Yes, it is visible.
I think it's a recorded version, and it has been started.
Yeah. Can you play this, Dr. Roy?
Yeah, it's already started actually.
Okay. We'll wait for a second.
Yeah. Can you all hear?
I think the volume has to be a little bit more, Dr. Roy.
Yeah, I think as it is a pre-recorded one, so I don't
Okay.
have any ability to increase the volume from here.
Oh, okay. So sorry to hear that.
Yeah.
Dr. Ekach, can this be done at your end if it's possible, and we can go to Roy otherwise?
Ma'am, Dr. Ekaj is in another meeting actually,
Oh,
and he
okay.
has requested me to take it over.
Oh, okay. I'm just trying on my end if I can get it working or if the volume is better. Dr. Roy, do you want us to go to your this one till then, or we can just play this?
I think, ma'am, do you want? Okay. Can you hear me?
Yes, I can hear you.
Oh, okay. So will you try from your end?
I am trying till then. If you can go ahead and have your session, then we can go to Dr.
Sure,
Mushtabaz at the last end. Okay. So I'm just
Okay,
going
great.
to, yeah.
Yeah.
Can you just share the panelist this one again with your slide? So I can just
Yeah, I'll
introduce
do
you. And until then, I'll try and make this one work. So if it is working, we can have this up and running. Thank you. Sorry for
Okay.
these. Yes, Dr. Roy, so you can share the panelists for plasma protein, the Google slides to your this one, and then I can do the next one.
Okay, ma'am. Do you want me to share the panelist slides?
Yes, please, so
Okay,
that I
sure.
can introduce you
Very-
and then you can go
Oh,
forward.
sure, sure. Is it visible now?
Yes, just go to your this thing,
Yeah.
Dr. Roy. So most of us actually already know Dr. Kalyan Roy. He's from Bangladesh. And Dr. Kalyan Brother Roy, he is DBS and DBS and T, Clinical Staff Transfusion Medicine Unit from Square Hospitals Limited, Dhaka, Bangladesh. He has completed his education under Bangabandhu Sheikh Mujib Medical University. He has 8+ working experience on transfusion medicine and 2 years of teaching experience. He's also a member of BTSB, ASFA, and ABB. I know Dr. Roy as a resource person for academic activities at ATM. So it's really good to have you here, Dr. Roy. And we can go to your next questions, please. So these are the questions that I had posed. And even if you may not have fractionation in Bangladesh at this point, but what are the challenges that you're looking at? What would you require to comply with these requirements? So Dr. Roy, if you can present your presentation, we can look into that. Thank you so much.
Yeah, thank you, ma'am. Am I audible to all?
Yes, you're audible.
Just give me a few seconds to share my slides, then coming.
Yes, you can share your presentation now.
Yeah,
Thank you.
ma'am. Can you see it, ma'am?
Yes, we can see it. Thank you.
Asha ma'am, can you see it?
Yes, yes, yes,
Oh,
we can.
okay, okay. Thank you very much for inviting me as a panelist in this elite group. And regarding the first question, I'd like to say that right now we don't have any fractionation center in our country, but we think we need a fractionation center for this huge population. And I have a presentation and that will actually give a little bit of overview of current situation of the plasma fractionation and the BTS system country. So Bangladesh is the most country in the world, and the current population is more than 167 million. HIV/AIDS prevalence rate is less than 0.01%, and in 2020 GDP was 324 billion USD. So the BTS has actually divided into two divisions, like the government sector and another one is the private sector. So we have 247 government BTS and private sector 145. The blood donation status of Bangladesh, this data is collected from 2018 to 2019, and there were blood donation of about 974,000 units. And we have mostly two types of donors. One is the BNRD, or the voluntary non-remunerated blood donors, and another one is the replacement donors. So we also have the blood strategies, and we had 30% of the blood components in 2018 and 19. And the TTS screening status of Bangladesh, TTS screening is mostly in the method, and only few some centers screening is done by method. And Bangladesh maintains a very low prevalence, less than 1% of the TTI markers. So apheresis and blood component therapy. Component facilities, we have 42 centers, private and government, and 52 centers, private and government. And we do have the facilities of different types of apheresis. We go for plasma, platelet apheresis, plasma apheresis, therapy plasma exchange, stem cell apheresis, erythrocyte apheresis, leukocyte apheresis, and therapeutic cell exchange. So surplus plasma, though there is no exact data on surplus plasma we have annually, but as only 42 centers has component preparation facilities, rest of the BTS are unable to process these FFP. And this table is showing that 785 units of the plasma discarded in 3 years in a tertiary hospital where FFP storage facility is available. So the PDMPs we import are the human albumin, IVIG, hepatitis B immunoglobulin, NTD immunoglobulin, coagulation factor 8 concentrate, and coagulation factor 9 concentrate, rabies vaccine. And the clotting factors, these are mostly donated by the World Federation of Hemophilia through its humanitarian aid program. What are the challenges we are facing to have a plasma fractionation? Number one is that the inadequate blood donors, lack of estimation of PDMPs, and lack of the demand from the physicians to use the blood components, lack of component facilities in tertiary and district hospitals, and lack of skilled manpower, and regulatory and legislative issues, and GMP and QC management problems. But there is definitely a light of hope. And what is the most important thing for us is that we have the powerful equipment that is our manpower or our population. So there are a lot of potential plasma donors, and our government is thinking, and soon the government will take necessary steps to go for a contract fractionation or maybe establishment of plasma fractionation in the country. So this is all from my side, and this is the update from Bangladesh. Over to you, Dr. Asha. Thank you.
Thank you so much, Dr. Kalyan. Very nicely presented. And I think, the things that all of us have spoken, even Dr. Hippie, Dr. Chandani earlier, they have mentioned and they have spoken. I see a common thread that is for all of us to, really explore and how we can help each other overcome some of the situations. And as Dr. Heen also mentioned during her slide decks, are we the lower income or the medium income countries have to come together and explore how we can look into various modalities. If there is a fractionation unit or a fractionation company, how we can help each other and provide support to the patients. And I think Doctors Beyond Boundaries is a very good option, and I think we should really look forward on how. So I again really thank AATM for providing this platform, because through this platform, I have learned so much more, because each other's situations, how we can, overcome it. So although I was trained in a rural hospital, but, how we can overcome that is very important. I think I can make the last one work. So I will try and play Dr. Mustapha's. I have already introduced Dr. Mustapha's sorry, I've already introduced Dr. Mustapha. I'm just going to make this work, if I can make it work. Just a second, and my apologies for having multiple windows open, but yes. Let me know when you can see it. Can you see this? Can you see the screen?
Yes, ma'am. The screen is on.
Yeah. So I'm going to start now, okay? And
Is it possible to make it slideshow?
This is a slideshow with the voice message actually, and the voice is not coming so much loud.
sorry, Dr. Roy, can you just say again?
Well, what I have observed, ma'am, in this slide, there is a voice message from Dr. Mustapha, but the voice is not so much louder.
Okay, I'm just going to play it again, and my apologies if you just 1 minute, just one
Sure,
minute,
ma'am.
Dr.
Sure.
Roy.
I think the slides are not working, ma'am. Uh, is it audible to everyone?
No.
Okay, then oh, the slide is over right now. So I'd like to introduce Dr. J.P. Prasad sir, and I would request him to share his views on the plasma fractionation. Sir, would you be kind enough to tell something about the plasma fractionation and your experiences?
Okay. Thank you, Dr. Roy, and thank you, Dr. Asavari. And I am sorry I could not send my presentation due to some little confusion. I am audible?
Yes, sir. You were loud and clear.
Okay. So I am sorry this time, but I am Dr. J.P. Prasad, and I retired in 2021 December from the post of Deputy Director Quality Control. Our institute, National Institute of Biologicals, is the only institute who is doing the quality control of the biologicals, like plasma-derived product, human albumin, factor 8, factor 9, immunoglobulin, fibrin cell and kit, recombinant product like insulin and erythropoietin, then vaccines. So I look after the most of the time of the plasma-derived product. So the presentation and the management of the Asavari is the very good. As a moderator, she has played a very important role, and she has presented well. And other participant also, our this presenter, they have presented well and give the insight about the plasma-derived product. And most of the people are from the transfusion medicine. So just I will give my experience about the quality control of the plasma-derived product. As Dr. Hina, she has said in our presentation, the albumin, IgI, and factor 8, and she talked about the sodium content of 130 to 160%. As per my experience, if any country, they do not have the quality control testing facility, so you cannot believe the quality of the product. So in 2016 and 15, I have failed total 29 batches of the albumin on the basis of sodium content. As per pharmacopoeial standards, there should not be more than 160 mole, but I found during testing more than 200. Suppose this product released in the market without testing and transfused to patient, of what the consequences. So the quality control is the very important parameter, and we are doing the total quality as per the Indian Pharmacopoeia monograph. So not only the sodium content and potassium or stability of protein is important. Important is the pH. Important is the molecular size distribution. Important is the pre-calculin activator. There are many more parameters that is the more important. Reason behind that, many a time I have failed on the basis of simple test pH. And I will give one example. There is a very good company and the reputed company worldwide that is from the Germany. I cannot take the name as a confidential, but I will just explain the whole history of that. We got imported product by one company in India, 2 batches of that product, and I fail on the basis of protein content, right? The protein content should not be more than 20. If the protein is 20%, that should not be less than 19% and not more than the 21%. When I tested in our laboratory, we found that was 22%. So we fail on that basis, and the manufacturer vendor, they complained case are in our laboratory, Paul Eldrich, Germany, they have released the batches. So our product is good. I'm like, yes, I know your product is very good. There is no doubt about that. But as per my test, your product has been found not of standard quality. Then I requested them, kindly send your standard, whatever standard you have used to test your protein content. Then I have tested their standard as well as their product. Whatever they are reporting, 21 point, 20 points some protein, then I got the same result with their standard. When I put our standard, we got more than that. So that means there was some problem, and we put other manufacturer put in also albumin also, and all are including my standard fail on their standard. Then I studied their standard. They are saying that my standard is of 5%. But when we tested in our laboratory, that was find 5.47%. And then I requested to the one scientist from the Paul Eldrick, Germany, and I requested for the standard. They send the standard. I test in our laboratory. Then whatever you have, they are reporting for their standard, I got the same result. So I confident enough that my procedure is okay, my standard is okay. The problem is with the standard of that company. So I requested them, kindly change your standard. Then they have changed their standard. This is the one story of what standard we are using for the test analysis. One, just I am in 2002, I have failed one batch of National Institute of Standard Biologicals Control, UK standard, poly-specific AHG, poly-specific AHG standard. On the basis of that, they have accepted our result. So we have tested their standard also, and that was not found okay. And they accepted, they accepted happily. So this is the one reason. Second one is the pre-calculin activator. We have imported some product from the company. The pre-calculin activator should not be more than 35%. But when we tested in our laboratory, that was found 55. And they have also tested in our laboratory, and they exported in India. They might be knowing that India is the very, it is not scientifically high-fi country. That's why they have sent it. Then I asked the manufacturer, you are knowing that your product have 54 unit of pre-calculated. Why you have sent exported to India? Then my one scientist told, sir, they took the chance. If the India release that product, and you might know that the region, what the excess of pre-calculin activator cause this dilation of the venous, then decrease your blood pressure. So these are the regions. So that's why the quality control of the plasma derived products is very important. Do you have time to listen, or you want to?
Yeah, Dr. Prasad, I'm so sorry. We are I think India 4:30 p.m. and UAE 3 p.m. But I think looking at overall, and I would request all panelists to open their mics if they feel like talking or if they want to, because I enjoyed, this was a good discussion, although this was not what I signed for, Dr. Sangeet, when I accepted that I'll be the moderator, but it has been a very enjoyable experience. And I think looking at it, what I feel, Dr. Prasad, is some of these comments that you have made, why don't we have another round of discussion? Okay. And I'd like you to come back with a presentation and some of the data that we have presented today. I think we should, write a publication on behalf of, AATM, so that we can publish this data, because all of us, when I say Dr. Chandani's comments, the way she presented her Sri Lankan experience, Dr. Nova, Dr. Hina, Dr. Subrato, and you. So, when we look at all these perspectives, I think, and unfortunately, Dr. Mustapha's we couldn't, but what I'm going to do is, I'm going to ask Dr. Aikaj to share it with you all on our PPT, in our group, if that can be shared, or Dr. Kalyan, if you can share that, but of course, take the permission from Dr. Mustapha. I think he should be okay because he has already provided us with a sign consent. But yes, at the end of the day, what I feel is, I just want to be mindful for everyone because it's over the hour. And actually, we were thinking we may not have so much to discuss. So I'm happy that we have
I want to add one thing to Dr. Hina's slide. she has showed that the 6 to 9 decigram per DL protein. So I think that is 6 to 8 or 8.3. She can check from where she has taken. She can check if she give the reference. Right?
Sure.
But that's okay.
Sure.
Thank you very much. Thank you.
And
Next time,
the 9 gram per deciliter is taken from the CFR standards FDA.
CFR standard FDA. Okay.
Yes.
Thank you.
Yes.
Thank you.
This is the reason why if we can have another round of discussion, I'll be very happy. I will, get to more, details on, what we are doing, how we are doing it. And I think at the end of the day, how we educate each other, whether through a workshop, whether through these seminars, is very important, because then we're trying to say that, okay, this is how it is. And I think, Dr. Prasad, just something that I want to say is, I think, when I started 20 plus years back, of course, I'm junior to you, but I have realized that it's people like you, stalwarts like you, who have worked hard in this field, that we now, whether we are Indian, Pakistani, Omani, Emirati, any one of us, or Bangladeshi, I mean, all us Asians, basically, now when we go abroad, we are provided with that value, because now they understand that, yes, there's a lot of data coming in from our countries. And I think all of us have to learn, if we unite together, and it might sound very political at this point, but without anything, scientific, doctors without borders, if we come together and provide this, this thing, we can help our patients, we can help our communities. And I feel very strongly about that. So, thank you so much for bringing this forward. I think, Dr. Kalyan, you have your hand raised. Please go ahead.
Thank you, ma'am. I have a question to JP sir. Sir, do we need net testing for plasma fractionation? Is it necessary to have a net testing?
So far as net testing is concerned, only we have to test for the SCB, RNA net testing. That is required. Otherwise, so far as HIV1, HIV2 or HBsAg concerned, need not to test, because we have the robust manufacturing system, and we have 2 steps in manufacturing process to remove or inactivate the viruses. Suppose we are fractionating by the alcohol, that alcohol is also, and downing the pH is also important to kill the virus or inactivate the virus. There is some filtration in factor 8 or heat treatment. So there are minimum, as per guidelines, there should be minimum 2 steps which remove or inactivate the viruses, and that increase the safety of the product, plasma derived product, right? So, so far as net testing is concerned for the all viruses, that is not necessary. We have the robust system. Okay, Dr. Kalyan?
Yeah, thank you, sir. And another question, the last one is, can we
Dr.
produce PD?
1 minute. We are all connected in the ATM PPT, group. So please feel free to refrain. And the reason why I am, putting a, strong this thing is because we should be mindful of each other's time. I want Dr. Prasad to come back again and not, share it just with a few people. We want 300 people on this presentation next time. So we will work hard towards that, and we'll get more towards.
Yeah, madam. Thank you, madam. Actually, there was some little confusion with me. That's why I cannot send my presentation. Otherwise, my presentation and everything were ready. So next time, there is
Definitely. I look forward, and I'll connect with you as soon as today's session is over. Dr. Ananda, can we request you to just say a few words if you're free or available, or Dr. Sangeeta, anyone?
I can see Dr. Anand Asawari. I'll wait for his comments.
Sure. But I think overall, it was really nice. Please keep the questions going, and we are there on the group. We're already having quite a few ideas. I want Dr. JP Prasad to come back again and learn from him. And I think even Dr. Hina, thank you so much for this presentation. Dr. Chandani, Dhamika, Dr. Nova Hippi, Dr. Mustapha, although we couldn't hear him, and Dr. Kalyan Roy. I really thank you. Although this wasn't what I signed for, because I'm in the middle of my vacation, but I had to, work to get the basics done, but it was enjoyable for me, and I would like to come back as a moderator if Dr. Sangeeta and Dr. Ananda would agree to that. Dr. Sangeeta, I think you go ahead and say your provide your
Yes. Thank you so much, Dr. Savri, that you took the lead. And it was actually very interesting to hear from each of you, from each of the countries, the different aspects of plasma collections, quality controls. And as you said, yes, we can compile all this and come up with a good paper. And that is a very good insight that you've given us. And like it was planned in a very different way, but it went in a different way. But I suppose it was really good. So thank you so much, each one of you who were present here. And we shortly will come up with another plasma, discussion, panel discussion, or any webinar type on a plasma protein therapy group, and will involve each of you who were present here today with us. So thank you so much for your time. And Savri, thank you so much, especially that you came for us out of your vacation. So thank you all so much. Thanks.
Thank you so much. I think half of it goes to my husband and my son, but overall it, they lasted me. They told, they were okay with me going through. But again, thank you so much. And I could also see Dr. Meena. Dr. Meena, she's from Iran. They are also doing plasma fractionation. So I would encourage her to come back next time, and we'll be very happy. There are two things which I want to, discuss with our participants and panelists today. One is, every one of you did an excellent job today and presented your own countries and what you feel is important and the challenges that you face. If you can send all of that documentation to me, Dr. Sangeeta, Dr. Anand, and Dr. Kalyan, we could try and come up with, or anybody wants to head this project, let's head this project and write this down as a paper, because I think we had very good material today. And this is exactly what we need to write down and document and publish it. And second time, if you want to speak again, get in touch with us so we can organize it better with either a 2.5 hours of workshop with Dr. JP and some other discussions that we can have, and people would actually gain a lot of value on how to implement these programs. So I'm going to leave you with thoughts. Yes, please, Dr. Ananda. No, I think he's coming in and coming out. So
yeah, there's some connection problem, I suppose.
I know, I understand. But I think this is why it was enjoyable. And thank you everyone again, and hope to see you soon. And we'll be very happy. I think once Dr. Ekaj is out of his meeting, we'll be able to share the recording as well as Dr. Mustapha's presentation. Thank you so much, and a big namaste from me. Thank you.
Bye-bye. All bye.
Thank you, Dr. Sangeeta.
Thank you so much. Bye, everyone.
Bye-bye.
Thank you from Bangladesh. Thank you, ma'am. Thank you, sir.
Bye.
So I'm going to wait till everybody goes away. Then I think freezing plasma is very important for keeping coagulation factors. I can see, Doctor. We will get these comments,
Yeah,
Dr. Roy. Yeah. So if anybody has a question, we can put it in the PPT group so that people can answer. So Dr. Roy,
Asham, if you have the authority to stop the video or recording, then you can stop it right now. I think the session is over. I don't have the option right there.
Oh, okay. Now is it closed? Can you check? Sorry, I didn't
Not
know I
yet.
had the option.
Not yet.
Not yet? Okay. No, I didn't know I had the option. Just a second, sorry.